So now I'd like to kick off the talks of our retreat with Michael Werner, the first keynote speaker. Michael has just arrived at the University of Utah from the Max Planck Institute for Developmental Biology in Tübingen, Germany, where he's a postdoctoral researcher with Ralph Sommer. Prior to that, in May 2016, he earned his PhD in the Department of Molecular Genetics and Cell Biology at the University of Chicago in the laboratory of Alexander Ruthenberg. Michael was a Chicago Biomedical Consortium scholar and a recipient of university-wide Teaching Assistant Award. His research there focused on non-coding RNA, chromatin, and genomics. So Michael's current research combines his PhD and postdoctoral experiences in dissecting epigenetic mechanisms of phenotypic plasticity and their potential roles in ecology, evolution, and human health. And I'm sure he'll introduce you to his system, what he's done, and what he's planning on doing. Michael is a native of Palm Bay, Florida. During Michael's presentation, if you have a question for him, please enter it into the chat box. We will moderate the questions after Michael's talk, and we'll do our best to have him answer as many questions as possible. So Michael, I'll turn over the podium to you.
Okay. Can everyone see my shared screen? Okay.
Yeah, looks good.
All right, so thanks for the introduction, Neil. I just want to quickly say how happy and excited I am to finally be here at the University of Utah and to join the faculty at the School of Biological Sciences. I look forward to meeting everyone in person, hopefully in the not too distant future. But for the moment, I'd also like to acknowledge and recognize the work that the organizers put in to making this event happen today virtually. And so thank you for your efforts. And so without further ado, I'll go ahead and get started. So again, my name is Michael, and I'm going to tell you today about some of my postdoc work, which will serve as the foundation for some of the projects that I'll start in my new lab. So work from many labs over the last 25 years or so has shown that certain post-translational modifications, specifically DNA methylation and certain repressive histone modifications, as well as non-coding RNA, are required for maintaining cell identity or cell lineage. And importantly, some of these modifications can actually template a self-replication mechanism, providing long-term transcriptional memory well beyond the initial inductive signal. And this mitotically heritable change in gene expression that doesn't involve an underlying change in DNA sequence is referred to as epigenetic memory. And this epigenetic memory is required or important for maintaining a T cell as a T cell and a macrophage as a macrophage. Now, these are incredibly complicated processes, and I don't mean to imply that we fully understand them, but we have learned a lot. And I think we've learned enough that we can start to expand the scope of some of our questions. And the question that really gripped me when I started my postdoc was, are there similar epigenetic mechanisms affecting organismal phenotypes? Or phrased another way, what are the mechanisms of phenotypic plasticity? And the hypothesis is that they may involve the same mechanisms that regulate cellular plasticity. And so just to give you a little bit better idea of what I mean by organismal phenotype, the causes and consequences of this phenomenon, I turn to a genetic model system, specifically the nematode Pristionchus pacificus. I realize many of you may not be familiar with it, so I'd like to sell you on this great system. So Pristionchus shares many of the same advantages as the more classical nematode model C. elegans. It has a short, fast hermaphroditic life cycle. It can enter a dormant dower stage upon extreme stress. And it has a small genome, and it has many, if not all, of the same forward and reverse genetic tools. But the thing that separates Pristionchus from C. elegans and makes it really useful for the kinds of questions I'm interested in is that it exhibits phenotypic plasticity. So the adult worm can develop one of two possible mouth forms, either a stenosomatous morph, which is Greek for narrow mouth, or a wide, or a urosmotus morph, which is Greek for wide mouth. And in addition to the extra width of the buccal cavity shown here, the stenosomatous morph has one flap-like dorsal tooth, whereas the urostomatous morph has a more cuticularized and hook-like dorsal tooth and a grossly enlarged right subventrolateral tooth. And the opposing force of these two teeth can be used to grab onto other nematode, break their cuticle, and actually start to eat them. And so these urostomatous morph adults are actually predators, whereas the stenosomatous morphs are obligate bacteriivores. And so I know these Greek words can be hard to keep in your head, so throughout the talk, you might see me refer to them, the stenosomatous morph with this single blue flap-like tooth, and the urostomatous predatory morph with these two red teeth. Okay, so just to give you a visual of what predation looks like, this is a predatory urostomatous morph grabbing onto a juvenile, and I've stained the juvenile with a vital dye, which is in its intestines. And I think this is useful because you can see right about now it starts to intake some of the intestines of this juvenile. And so observations like this have led us to conclude that not only is this predatory behavior useful for killing competitors, but also as expanding their dietary range. And again, to remind you importantly, the stenosomatous morph does not exhibit these predatory behaviors. So it's a plasticity both in morphology and in behavior. And the reason that this is such a useful model system, I believe, is because the summer lab where I did my postdoc has spent the last 10 years decoding a gene regulatory network for malform development. We also know a little bit about the ecology of the worm. For example, specific small molecule pheromones that are produced exclusively in adults can induce the predatory morph in other individuals. And so we think that this is a sensor of crowding or soon to increase population size. And fortuitously, we found that simply manipulating culture conditions can induce one of the two possible malforms. So simply growing worms in liquid culture can promote a largely stenosomatous population. Conversely, taking those same worms or same eggs and growing them in agar can produce or promote a mostly urostomatous population. And you'll see shortly how this has become a really useful tool for us to understand the molecular mechanism of this switch. So getting back to the central question that I have, do epigenetic mechanisms maintain a record of environmental exposure in the form of organismal phenotypes? In the context of this specific system, is there environmental memory that influences malform choice? Is that memory dynamic? Is it regulated by putatively or potentially epigenetic mechanisms? And if so, which genes are they regulating? So to get at the first question of environmental memory, I took an approach that's somewhat similar to an old school reciprocal transplant experiment. And so I'm going to show you data on the y-axis of percent urostomatous or percent predatory of adults that have been switched between agar and liquid culture conditions at the indicated time points on the x-axis. And what we see is that in the first 24 hours of development, switching to the liquid culture condition elicits a low urostomatous population. So this is not significantly different from having grown the worms in liquid culture the entire time. And so this seems to be a complete phenotypic conversion, indicating complete plasticity in this first 24 hours. However, as we start to switch later in development, we start to increase the ratio of Urus predatory animals, which indicates a memory of having grown an auger prior to this developmental switch. And of course, we can perform the reverse experiment, and we see a reciprocal trend in the same sort of pattern. And so, is there environmental memory? I think this experiment, this very simple experiment, suggests that there is. There's a very early and a completely plastic phase of development, which is followed by a period of increased memory of the previous environment's condition and phenotypic preference prior to ultimate differentiation of the malform development. So it seems like there's environmental memory. Is it regulated by putatively epigenetic modifications? And so this experiment is very straightforward. We grew worms either on an agar plate or in the liquid culture flask, and we added a panel of chemical inhibitors to histone modifying enzymes. And what we found is that there's a drug, NU9056, which led to a primarily stenostomonas population, so low Urusmonas population compared to the control DMSO, as well as a drug called trichostatin A, or TSA, which led to a preferentially Urostomonas population compared to control DMSO. And the really exciting thing about this result is that NU9056 is a histone acetyltransferase inhibitor, whereas TSA is a histone deacetylase inhibitor. And so we have opposite phenotypes or opposite effects from correspondingly mechanistically opposite functions of these two chemical inhibitors. And so I think it's not to exclude these possible other modifications, but combined, this really strongly suggests that acetylation is important in malform development. And so to see if acetylation is important in the memory that we just observed from the switching experiments, we took the following approach. So we know that if we grow worms in agar and switch them to liquid culture at the early time point, we get complete conversion to the liquid culture phenotypic preference. But we wondered, if you block deacetylation by adding TSA, does that effectively prevent phenotypic conversion? And if so, I think this is compelling evidence that the acetylation mark established early in development is important for maintaining the memory of that early environmental exposure, which would potentially fix that developmental trajectory. And so this is the experiment we conducted. This data I'm showing now is the first data that I showed you, where we have % Urusmonas on the Y. If we grow worms in liquid culture and you add DMSO, you have a low Urusmonas population. If you add TSA, you get this sort of intermediate ratio of Urusmonas animals. And if we perform the switch experiment between agar and liquid culture, but then add TSA at that time point, we see a huge induction of Urusmonas animals, mirroring actually growing worms on agar for the entire duration of their lifetime. And importantly, not only is it different from the direct control of adding DMSO, but it's also statistically different from adding TSA at the beginning of the experiment, which suggests that there's a combined effect of the deacetylase inhibitor and the environment, which is consistent with our hypothesis of fixing that juvenile chromatin state. And we thought we would conduct a few experiments to try to refute that hypothesis. One idea is that adding TSA later simply has a stronger effect because of degradation from the earlier time point. And so we added TSA at 24 and 48 hours, but without this switch. And here we saw decreasing intermediate ratios of Urusmonas animals not able to recapitulate this full induction shown here. And then what I think is perhaps the most elegant experiment, we repeated the switch experiment, but only added TSA at a later time point, at 48 hours, so a full 24 hours after the switch. And again, we were not able to recapitulate the full Urusmonas induction. And I think together these experiments really strongly argue that there's a combined effect of the environment and the deacetylase inhibitor, and it depends on adding the inhibitor at the time of the switch, which when put together, I think really argues that the acetylation mark is providing a memory of that early agar juvenile state manifesting in that Urusmonas induction. Okay, so which specific acetylated lysine provides this memory? This is again a straightforward approach. We took worms, grew them in liquid culture with our control DMSO, our experimental sample TSA, this deacetylase inhibitor, and then we threw in another negative control, which I like a lot, which is sodium butyrate. This is another HDAC inhibitor, but it had no phenotype, so it serves as a negative control. We extract histones and then perform Western blots and liquid chromatography tandem mass spectrometry. So the Western blots showed us no noticeable difference in a histone 3 panacetylation antibody. So this is a non-specific antibody to any acetylation on the histone 3 tail. We saw no noticeable difference in TSA compared to the two controls, normalized to total histone levels and shown quantified here. And not surprisingly, because of this result, we also saw no difference in K27, H3 K27 acetylation. This, at the time, was surprising to us because we really thought that this would be the mark that's moving around and responsible for this effect, because it's a classical mark of enhancer. It's been shown to be involved in other examples of phenotypic plasticity, but nevertheless, we saw no difference. However, we did see about a twofold difference in histone 4 panacetylation in TSA compared to controls. This is one replicate. Of course, we performed it in triplicate, and we do see a nice statistically significant difference in induction of H4 panacetylation in the presence of TSA compared to control. Now, to identify which specific lysine residue is affected, we repeated Western blot, but with antibodies specific to the individual lysine residues on the histone 4 tail. And we're really nice and surprised to see that there's actually only one mark that responded significantly, H4K12 acetylation. There was modest increases as well in K5 and K16 acetylation, but they were not significant in this experiment. And so we were really excited about this idea of having a specific epigenetic mark responding to TSA and perhaps providing this memory. And so we wanted to verify this result with LCMS, which is, of course, a more sensitive instrument. And really nicely, we confirmed that K412 acetylation is the strongest inductive mark, more than 5-fold induction in this measurement. We also did see K5 acetylation become significant in this experiment, presumably because of the greater sensitivity of this measurement. And so combined, which acetylated lysines provide memory in this experimental setup, it seems to be H4K5 and K12 acetylation. And so then the task is to identify which genes are these marks presumably regulating. And so we took a transcriptomic approach. We performed RNA-seq on liquid culture or in agar. There are two environments. At multiple time points throughout development. And as one often observes in a transcriptomic experiment, you see hundreds of differentially expressed genes, shown here in total number on the Y at each of these different time points between the different culture conditions. And normally this is a conundrum where you have to figure out which genes to focus on. But here again is the strength of this model system where we already have an a priori known set of genes involved in malform regulation. And so we could focus on this list, and one gene in particular really stuck out, which is UD1. It stands for Urostomatus defective 1. This was the first and only gene to be differentially regulated at 24 hours, and it maintained that differential regulation up for 3 time points. It was the only gene in this regulatory network to have consistent differential regulation. And so this is showing you what that actual data looks like. This is gene expression on the Y, again, RNA-seq data, and time on the X. And so there's an induction of UD1 on agar, the Urostomatus promoting condition, somewhere between 12 and 24 hours. That does not happen in liquid culture. And then it decreases, but maintains that difference up to 60 hours of development. And so we know it's responsive to the environment sort of statically, but is it also important for this phenotypic conversion that we saw earlier? And so this experiment is performed using RT-qPCR. So the y-axis is again expression of UD1, and x is time. And the trend in blue is the abundance of UD1 mRNA we see in liquid culture. And now we thought we would repeat the experiment, but switch at 24 hours and see if there's an induction of UD1 to that auger sort of trajectory we had seen before. And indeed there was really quite a powerful induction very quickly within 12 hours, which is consistent with the phenotypic conversion to that to the auger phenotype. And then to combine UD1 regulation and histone acetylation, we simply added TSA in liquid culture and asked the question, does it affect UD1 transcription? So again, this is qPCR expression of UD1 on the Y, time point on the X. And we did see a statistically significant induction or enrichment in the TSA light blue bars compared to the DMSO control in the dark blue bars. And very interestingly to us, the difference was at 48 and 72 hours, not at the 24-hour period. And this is consistent with the idea that acetylation is not involved in the initial, or at least K4, H4, K5, and K12 acetylation, is not involved in the initial induction of the environment, but in its repression. And so that's all consistent with the idea of it containing memory rather than the initial inductive signal. And then to really put all of these pieces together, does H4K12 acetylation act at the UD1 locus in the memory paradigm that we've established? And so the data I'm showing you here is chip qPCR at the UD1 promoter, and the y-axis is percent of input. And so what we see is that between 24 and 48 hours, there's a decrease in the abundance of this mark at the UD1 promoter. We then perform our switch experiments, auger to liquid culture, without adding TSA, and at 48 hours, we see a similar low abundance of this mark, but now we repeat the switch experiment, but add TSA. And here we have a high level of K12 acetylation again, and this is basically maintained. The exciting interpretation here is that we've actually maintained the same level of acetylation that we saw at 24 hours, preventing its otherwise decrease. And this is very consistent with the phenotypes that we observed when we performed this experiment. And similarly, we check to see if adding TSA has this effect without the environment, and it does not. So again, there's the combined effect of the environment and the deacetylase inhibitor, and it locks in what appears to lock in that early chromatin state at the UD1 promoter. And so just to conclude, do epigenetic mechanisms maintain a record of environmental exposure in organismal phenotypes? I think the jury is still out to conclusively say this, but there's a lot of fun and exciting experiments, I think, to really get at whether this is happening. But I do think we can conclude a few things from the experiments I showed here today. One is that phenotypic plasticity does exhibit environmental memory in this system. Environmental memory is stored in H4 tail acetylation at a single switch gene, which is really surprising to us. And fixing histone modifications with a chemical inhibitor effectively freezes a juvenile chromatin state and developmental trajectory. And the implications of this result, I think, you can imagine beyond this experimental paradigm being quite interesting and quite broad. And so with that, I'd really just like to thank my postdoc advisor, Ralph Sommer, a technician who worked with me on the project, Tobias Loschko. He's actually going to come and help me start the lab for a year. And then also I'd like to plug that I'll be taking rotation students. I'm happy to discuss projects ranging from biochemistry all the way up to evolution. And I hope I was able to excite some of the first year students about these projects. And if there's any time, I'd be happy to take some questions. Thank you.
Okay, thank you, Michael, for that talk. We do have time for a couple of questions, and I've got one that was sent to me privately by Eric Jorgensen. Can you hear me?
I can.
Okay. And that is, Eric's question is 4K12 also methylated?
That's a good question. So there's a very recent paper in NSMB, I think it's recent as a year ago, that showed it does get methylated, and I think these were in human cells, but that's the only evidence for it being methylated. Otherwise, it's not known to be. So it seems like it is, but I guess it's yet to be, that study's yet to be replicated.
Okay, thank you. Austin is going to moderate some of the other questions that came in to everyone over the chat board.
Yes, thank you, Neil. All right, Michael, great talk, by the way. We do have a question from Lisa. Is N1 expressed in a particular group of cells? And if it's broadly expressed, could it control other organismal phenotypes?
So its expression is somewhat limited to a handful of neuronal cells. We have looked very extensively whether it affects other phenotypes. Broadly, the short answer is no. There might be some evidence that it has some role in oxygen sensation, and whether that was an ancestral function or a de novo function, we don't understand. But otherwise, these animals seem to be basically wild type.
Nice. So Fred has another question as well. He first said, great talk, and he thinks you had a nice takedown of C. elegans. But do you ever see intermediate adult phenotypes? Or do you see any interesting variation within the adult phenotypes?
So my internet is maybe a bit frozen. Okay, so yeah, can you guys hear me?
Yes.
Okay, so we 99.999% of the time there are no intermediate phenotypes. But in the most recent genetic screen, we did find a gene when knocked out, it gives you an intermediate phenotype. So environmentally, there seems to be very strong canalization or robustness to either the two phenotypes. And only genetically can we modify it. Now, that's in terms of morphology. We haven't really looked at behavior yet. And so that's one of the things I actually want to take a look at.
Awesome. And Neil, do we have time for one more question?
Yeah, sure. Let's go ahead with one more question. There's a lot of them on the chat board, and maybe Michael can look at some of those and provide answers to them, to the individuals. But yeah, let's do one more.
All right, so Vincent asks, so it sounds like the predatory condition is the one that needs to be induced through acetylation, meaning the bacteriivore is the default condition. Does methylation of the histone 4 protein prevent the induction of predatory conditions, or have you just focused on acetylation?
Well, we focused on acetylation because of that experiment where we added the different inhibitors, and we didn't see any strong effect with a methyltransferase inhibitor. And we looked in both conditions that promote either of the two malforms. So again, that doesn't mean we haven't found the right inhibitor or the right concentration or the right environmental condition, but that's why we focused on the acetylation.
Awesome. Great. Yeah, if you have any other questions for Michael, I would still just put them in the chat and they can peruse through them throughout the rest of the talks. But with that, Neil, I'll turn it over to you.
Thanks, Austin, and thank you again, Michael, for a nice talk and introduction to your research.